Just give me a second. My name is Gabriel Escalante, and I will be your host for today's session. Our webinar today is titled Imaging Hypoxia, and it will be presented by Dr. Katie Parkins, Head of Scientific Integration at Syntheca. Allow me to provide you with some background information about our presenter. Katie holds a PhD from the University of Western Ontario in medical biophysics. Throughout her research training, she was focused on the development and application of novel molecular imaging tools to monitor and treat diseases, more specifically cancer. Additionally, Katie completed a postdoctoral fellowship at Johns Hopkins University, where she used multimodal imaging to investigate the molecular mechanisms that may determine immunotherapy response. In her present role as the head of scientific integration at Syntheca, Katie supports our customers in understanding the products offered and how these instruments help to meet their research needs. During this webinar, Dr. Parkins will discuss the role hypoxia plays in disease progression and treatment response. She will dive into the various molecular imaging technologies that can be used to visualize and assess hypoxia in preclinical cancer models. Some modalities that will be covered during this session will be magnetic resonance imaging, positron emission tomography, and optical imaging. Before we begin, I would like to briefly mention a few housekeeping rules. We anticipate that today's webinar will take up the full hour, so we might not have time to answer your questions at the end. However, please submit your questions in the Q&A dialog box throughout the presentation. We'll create a transcript of the questions we do answer during the sessions, and we will, as well as those that we may not have a chance to address. We'll distribute this document within the next few days, and please also note that we're recording this session, and so if for some reason you lose connection or can't hear everything clearly, you'll have access to review the video file later. Without further ado, I'm going to pass things over to Katie.
Thanks, Gabe, for that introduction, and welcome to everyone who's joining us for today's webinar. To start off this morning, I'm going to give a brief background on hypoxia and the role it plays in cancer and tumor development. We'll then talk about the various ways this can affect patients and their response to various treatments. The majority of today's session, as Gabe mentioned, will be focused on the ways we can monitor hypoxia in our research, and specifically in preclinical cancer models. I'll briefly describe the technique while showing some examples of how it's been used and discussing the advantages and limitations of each method, in hopes that by the end of this, we can all take away a few new ideas of how we may be able to implement some of these technologies in our work. So what is hypoxia? While molecular oxygen plays a fundamental role in the human body, we know it's essential for developmental, physiological, and metabolic processes. Under normal physiological conditions, oxygen levels range quite a bit, so from about 1 to 20%, depending on the organ and tissue type. Low oxygenation may be physiological normal for some adult tissues which maintain a hypoxic environment. So this may be like, for example, bone marrow or thymus. However, commonly oxygen deficiency affects cellular functions and disrupts biological processes. So this can include things like proliferation, differentiation, angiogenesis, metabolism, and even pH. Therefore, hypoxia is associated with various pathophysiological conditions. This can include respiratory diseases, heart disease, metabolic disease, and notably cancer, which will be the focus of today's webinar. Hypoxia is a common feature of solid tumors. It develops due to the rapid growth of the tumor that basically outruns the oxygen supply and impaired blood flow due to the formation of abnormal blood vessels that supply the tumor. As a result, it's been reported that tumor hypoxia can activate things like angiogenesis, thereby enhancing the invasiveness and risk of metastasis. It can increase the survival of the tumor, as well as suppress anti-tumor immunity and hamper the therapeutic response. Now, in the clinic, patients with hypoxic tumors have been shown to have a worse prognosis and a reduced chance of survival. And this has been shown in a number of both preclinical and clinical studies, with hypoxic tumors showing resistance to therapy. And this includes both radio resistance and chemo resistance. As a result, a large focus of research has been geared towards developing hypoxia-targeted therapies. These can include pharmaceutical agents that directly target hypoxic tumor cells and the tumor vasculature, strategies that modify oxygen delivery to hypoxic tumor areas, and improved radiotherapy treatment planning coupled with hypoxia-specific imaging. Now, of course, in the clinic, for these hypoxia-targeted therapies to be effective, patients must first have hypoxic tumors. Therefore, identifying patients that will benefit from hypoxia-targeted therapies is really fundamental to improve the drug development. Now, in addition, it's important to monitor any early changes in hypoxia once a targeted therapy has been administered. Thus, there is a need for a robust, specific, as well as reproducible biomarkers of hypoxia, as well as reliable ways to evaluate them. While today's webinar will be focused mostly on imaging methods, I wanted to first review some of the more conventional non-imaging techniques that are used for assessing hypoxia in research, as I know many joining today may be currently using one of these techniques, and/or you may want to consider using one of them to validate your imaging studies. The first method is using needle electrodes. This approach was developed in the 1980s and used oxygen-sensitive needle electrodes to take direct measurements of oxygenation status at the tissue level. And this was considered a gold standard technique for accessible tumors. So things like head and neck cancer and soft tissue sarcomas. There's two different types of needle electrodes. There's Eppendorf and Oxylite. The Eppendorf electrode exploits the electrochemical properties of oxygen and produces a histograph of essentially regional oxygenation status, while the Oxylite, which is shown here, is a fiber optic electrode that measures fluorescent pulses that are inversely proportional to oxygen tension. And I'll show an example of this later on in the webinar. Now, while this strategy is widely used in preclinical cancer models, as well as other disease models, the technique is limited to clinically accessible and superficial tumor sites. Secondly, it may not accurately reflect tumor hypoxia across the whole tumor, and it takes around 1 to 2 minutes to acquire a measurement. And so this limits the number of measurements that can be taken accurately across the entire tumor site. Another established approach is to use histological staining for monitoring hypoxia. This method is reliant on either endogenous or exogenous biochemical markers. And so endogenous markers of hypoxia are proteins whose expression levels may be upregulated or altered in hypoxic conditions, such as HIF1 and its downstream signaling targets. And then exogenous markers are bioreductive chemical markers that undergo reduction reactions at these hypoxic sites. The strengths of using histology methods for hypoxic marker examination is really that the geographic regions and microdistribution of the relative O2 can be studied. And so this allows further observations such as highlighting areas of proliferation or neoangiogenesis, also observing any co-localization or associations between hypoxia and other tumorigenic adaptations. Now, of course, there are limitations. Histology requires tumor tissue biopsies, which are invasive, and again, only applicable to accessible tumors. The tissue specimens collected measure hypoxia in a subsection of the entire tumor, and so it may not be a true representation of the whole tumor mass. And of course, serial sampling through repeated biopsy is not often performed. And so histological methods do not necessarily capture the dynamic aspect of acute changes in tumor hypoxia over time. And so as an effort to try and overcome some of these limitations, researchers have more recently developed non-invasive methods to measure hypoxia using advanced imaging technologies. These methods include positron emission tomography, or PET, and the use of hypoxia-specific radio tracers, magnetic resonance imaging, or MRI, and oxygen-sensitive pulse sequences, optical imaging methods that rely on imaging reporter genes that are responsive to hypoxia, and more recently, live cell imaging, which allows researchers to monitor and image cells within their desired hypoxic environment. So we'll start off with PET. PET imaging is routinely used clinically to diagnose and monitor the progression of numerous diseases, including a wide variety of cancers, but is also commonly used in many other areas of research, including to assess hypoxia. To image a certain molecular target using PET, a radio-labeled imaging agent is administered to the subject. Typically, this is done through an intravenous injection. The imaging agent can be a small molecule, peptide, engineered protein, or a nanoparticle, but it needs to be specific and selective for the target of interest. Now, as radioisotopes decay, they emit a positron, which then travels a short distance and collides with an electron in the surrounding tissue. This interaction causes an annihilation event, resulting in the release of two gamma photons in approximately opposing directions. Following the detection of gamma rays by the PET detector, the location of annihilation events are calculated by observing multiple events, and then the resulting signals are converted and reconstructed into tomographic images. And so these images will reflect the distribution of the imaging agent in the subject and hence provide information on whatever biochemical event the agent was targeting. Okay, so how does this work for hypoxia-based imaging? Well, hypoxia PET radiotracers should exhibit a number of properties. They should be specifically and irreversibly retained in hypoxic regions only and not in normoxic or necrotic regions. This is to ensure there's good signal to background contrast. The radiotracers should possess suitable chemical properties to diffuse and distribute freely and homogeneously into tissue in a non-specific manner, and also show fast clearance from well-oxygenated regions. They shouldn't be affected by other biological parameters such as perfusion or pH, and they should be cleared via the systemic circulation. Finally, radiotracers should be robust for monitoring tumor hypoxia changes and be reproducible across multiple tumor types. So with this in mind, the first tracer we'll talk about is FMESO. This is a first generation tracer, which has been extensively studied both clinically and preclinically, and is arguably the most commonly used PET probe for imaging hypoxia. It's highly lipophilic. It's able to diffuse readily into tissues and is only retained in regions of the tumor with less than 10 millimeters of mercury, making it highly specific to hypoxic oxygen tensions. It has been used successfully both in preclinical and clinical studies as a prognostic marker and can be used to select patients that would benefit from more aggressive and targeted therapies. So on the right here, I'm showing an example of this where mice bearing a human cancer cell xenograft were administered a single dose of drug, either 0.3 or 1 milligram kilo dose saline for the control mouse. 3 days after the treatment, mice were injected with FMESO, and then this accumulates intratumorally. So we can see here, you can quite nicely differentiate these signals. So the control mouse has quite a lot of hypoxia within that tumor region, whereas the treatment groups do not. There's a significant reduction here and hardly any in the highest dose of the drug. And they're able to quantify that and show that those are significantly different. They're also able to compare these to histology markers of hypoxia, and they show a really nice correlation here. They're using muscle as control tissue, and they're able to show the same thing when they quantify this out, where the treated mice have significantly less hypoxia in the tumor compared to untreated mice. There's a couple different ways that this imaging method can be used. First and most common is just comparing the ratio of hypoxic versus normoxic areas within a tumor relative to tumor size. And so here's an example where they use FDG PET to visualize the tumor, and then FMESO is used to exploit areas of hypoxia within that region. And of course, here we see that the area of hypoxia increases over time. Secondly, this technique can be used to guide hypoxia-targeted treatment. So if we can image hypoxic areas with high specificity, we can then determine which areas to target for these therapies, knowing that the target may change over time as the tumor grows or starts to respond to treatment. And this can all be monitored non-invasively with imaging over time. Alternatively, FASA is a second generation nitroimidazole radiotracer and has been used in a number of preclinical and clinical studies. It is more hydrophilic or water soluble, which improves clearance from normoxic tissue. and it's less lipophilic, reducing uptake in lipophilic tissues and leading to faster uptake in hypoxic tissues. Consequently, it has better washout properties, and this improves the radiotracer's specificity to hypoxic cells and increases the hypoxic to normoxic contrast, and ultimately reduces the post-injection optimal imaging time. Here I'm showing an example where a group from Denmark wanted to explore the use and essentially the reproducibility of PhasAPET as a reliable tool for assessing strategies that target or modify hypoxia. And as you can see here, the intratumoral distribution of the tracer was quite stable. So even though these are a day apart, they image again and they get quite a strong correlation when they do a voxel by voxel comparison of the two scans. There are limitations that need to be considered when measuring and monitoring hypoxia with PET. In addition to the slow tracer clearance that we talked about, PET images are constructed in relatively low spatial resolution due to the large voxel size. The large voxel size may confound the overall interpretation of the hypoxic volume, because voxels can ultimately contain normoxic, hypoxic, or necrotic tissue, and partial volume averaging can skew the analysis. And then, of course, as with any PET study, the infrastructure and cost should be considered, as PET is relatively expensive compared to other imaging modalities and requires access to a nearby cyclotron to be able to generate the required tracers. Okay, moving on to MRI. MRI is a fairly complex imaging technology, but we'll try to give a brief overview for those of you who aren't familiar with it. While all systems can be a little different, in general, an MRI scanner is comprised of a set of embedded coils. You have one coil that generates the main magnetic field, gradient coils that are used to localize the source of MR signal, and finally RF coils, which excite the sample and detect the resulting MR signal. The magnetic field from the MRI interacts with certain atomic nuclei in the body or elsewhere that have special spin characteristics and magnetic properties. Most often we use hydrogen atoms since they're abundant in living subjects, particularly in water and fat. Now, usually these protons are facing in random directions, but when a subject is placed inside the magnetic field, a significant portion of them align in the same direction. For the next step, a radio pulse is emitted. This interacts with the protons, essentially turning them so that they no longer align with the magnetic field. But as the radio frequency only happens for a moment, the protons will relax back to their aligned state. And as they relax, energy is released, which can be detected by sensors in the MRI machine. And then through some calculations that are beyond the scope of this webinar, the computer can determine what the tissue looked like depending on this energy that is released and show us an image of the tissue. Now, in terms of hypoxia studies, dynamic contrast enhanced MRI, or DCE MRI, sometimes also referred to as permeability MRI, is one of the main techniques which calculates perfusion parameters by evaluating T1 shortening induced by usually a gadolinium-based contrast agent passing through the tissue. Observing and quantifying the time course of this enhancement is really the major goal of DCE imaging. As a general principle, the degree of contrast enhancement depends on things like regional blood flow, the size and number of blood vessels quantified by their surface area per unit mass of tissue, and their leakiness or permeability. And while typically it's not possible to separate these components individually, by mathematical modeling, we can actually measure their combined effect reflected in the so-called transfer constant, or K-trans. K-trans is an index characterizing the diffusive influx of GAD contrast across the vascular endothelium, and it's become increasingly important in the evaluation of tumor response to different therapies, especially for non-cytotoxic agents that target things like tumor vascularity. Now, in addition to K-trans, additional tissue-specific parameters can be estimated from DCE, including the volume fraction of the extravascular extracellular space, the volume fraction of plasma, and then the rate constant for efflux of gadolinium back into plasma from the tissue extracellular space. Now, here on the right, this study investigated the potential of a drug to decrease tumor hypoxia and the ability of DCE MRI to monitor drug-induced changes in tumor hypoxia. And this is quite a common application for this type of MRI. This was in patient-derived xenograft models of carcinoma of the cervix, and they report that treated tumors show less abnormal microvessels, lower microvascular density, and higher fraction of hypoxic tissue, or higher K-trans, compared to untreated tumors. And they note that the higher K-trans of the treated tumors was most likely a consequence of increased vessel blood flow. Now, similarly, the purpose of this study was to investigate whether a different drug may induce changes in tumor microvasculature and hypoxia in 2 different models of pancreatic ductal adenocarcinoma. The analysis of the tumors revealed that untreated tumors showed higher K-trans than treated tumors, further supporting that low values are associated with poor tumor vascularization and extensive tumor hypoxia. Now, they also used histological staining to compare the levels of hypoxia between these 2 models. And they found that in one model, shown here, the drug significantly increased hypoxia, while in the other model, no difference was seen between treated and untreated tumors. And this is of importance because it supports what was seen in the in vivo imaging data, suggesting that DCE MRI may actually be an adequate measure of hypoxia for both treated and untreated tumors. Oxygen-enhanced MRI, or OE MRI, is an imaging technique which uses inhaled molecular oxygen as a contrast agent for signal enhancement. So for this technique to work, a high concentration of oxygen is delivered at a set time point. And when it's delivered, it dissolves within arterial blood and tissue plasma and causes longitudinal relaxation time to shorten and the rate to increase. And so the changes in R1 can be measured as a proxy of tissue oxygen concentration, with an increase representing normoxic tissue and a stable or slight decrease in R1 indicating hypoxic tissue. And so this allows us to assess the delivery of oxygen around the body to tissues and to tumor sites. Now, importantly, since this method simply uses medical oxygen and standard MRI equipment with no investigational tracers or devices, it's readily translatable between animal models and humans. So people on the research side of things really appreciate this technique for their work. So here's an example where they were studying a subcutaneous glioma xenograft. Animals underwent OE MRI followed by DCE MRI and then histological staining, seen here on the bottom. The area under the curve, or the AUC, was measured for the R1 curve for OE MRI and the gadolinium concentration curve for DCE MRI. And what they found was that changes in AUC were regionally heterogeneous across all tumors. Tumor regions with low perfusion tended to have a positive correlation with hypoxic regions and a negative correlation with vessel density. And then importantly, they evaluated the relationship between MR measurements and histological measurements of tumor hypoxia and demonstrated strong correlation. So once again, support that this is a valuable marker for clinical investigations. The limitation associated with using MRI for monitoring tumor hypoxia, and more specifically, I guess, for DCE MRI, is that oxygen tension measurements are affected by a range of factors other than oxygen delivery. So these can include hemoglobin saturation, vascular geometry, and consumption. So therefore, this technique may only provide estimates of tissue perfusion and the hypoxic nature of the tumor. And of course, for preclinical studies, many of us know MRI in general can be quite costly and fairly time consuming, depending on the type of data that you need and from which part of the body. With that, I'm going to pass it back to Gabe for a quick poll question.
Thank you, Katie, for the great discussion so far. Before we continue, we would like to ask a quick poll question. So the question is the following. Just give me a second. The Question is the following. Are you currently measuring hypoxia in your studies? Please select the option that applies to your case, and we will wait a few moments to let everyone answer the questions. We'll just wait a few moments and we'll continue with the presentation. Thank you for answering the poll questions, and I will now hand over the presentation back to Katie.
Thanks, Gabe. Okay, so we're going to move on to optical imaging. And when I say optical imaging, I know it can be used quite interchangeably for many groups. So I'm referring to bioluminescence and fluorescence imaging. We'll start with fluorescence. So for fluorescence, following the administration of a fluorescent imaging agent, so a fluorophore or a fluorescent protein, an excitation light of an appropriate wavelength is used to illuminate the subject. In the case of using a protein such as GFP, one must first engineer the cell line of interest to express GFP, or you could purchase a transgenic animal already expressing the reporter gene of interest and then illuminate the subject. This leads to excitation of the fluorophore or fluorescent protein and the subsequent emission of light. The light is then detected by a CCD camera. It gets analyzed and converted into an image detailing the location of emitted light. For BLI, it's quite similar. First, a small animal model needs to be developed that's capable of expressing or secreting one of the luciferase enzymes. And again, this could be a transgenic mouse or a mouse that's been injected with cells that can express or excrete luciferase. Second, the appropriate substrate needs to be administered to the animal, and this will lead to production of light from the enzymatic oxidation reaction of luciferase with its substrate. And from there, it's the same. So the light will get collected by the cooled CCD camera and an image is produced. Now, unlike fluorescence imaging, bioluminescence does not need an external light source. And because of this, tissues don't exhibit, and because tissues don't exhibit endogenous bioluminescence, greater sensitivities and higher signal to background ratios can be achieved compared to fluorescence techniques. So this is something to be considered as we go into some examples. Okay, so what does this look like in terms of hypoxia? Well, the most common way people are using optical imaging, I would say, is engineering their cells to be responsive to the hypoxic response element. And so if you think of a cancer model, for example, you would typically engineer them to express your imaging gene, whether it's luciferase or GFP, et cetera, and that would be constitutively turned on. So it would be on all the time. In this case, they put a hypoxia response element promoter in front so that only under hypoxic conditions, this imaging gene gets turned on. From there, you would continue on with your model as usual. So you would inject your cell line to generate the cancer model, and then you would image them. And when you image them, only hypoxic cells are going to produce any optical imaging signal. I'm going to show an example just because it'll be a little bit easier to understand this. This is a group out of Johns Hopkins that tried to implement a theranostic strategy to combine hypoxia imaging with treatment. And so they have the hypoxic response element in front. It is in control of both the luciferase for imaging purposes and also a cytosine deaminase prodrug, activatable by prodrug therapeutic gene. And so here they can show in vitro that it's working with a hypoxia mimetic. They induce hypoxia and they show with the substrate that the cells are expressing luciferase. In treatment, you can see the cell line has the therapeutic gene. When administered the drug, this signal decreases compared to this one. In vivo, they're able to show this as well. So before any drug is administered, they're able to monitor tumor hypoxia with optical imaging. So again, only hypoxic tumor cells are contributing to this signal. And then once they administer the drug, they show that this signal is significantly less than the animals that don't receive any drug. So this is quite exciting. This is kind of a unique approach where not only are they using the hypoxia element to image, but they're using that as a target for treatment. And they're able to essentially monitor the treatment with both, which is known as a theranostic strategy. Alternatively, if you didn't want to engineer your cells, in this work, they reported a water-soluble and stable phosphorescent macromolecular probe for hypoxia imaging with high sensitivity and specificity. And so the probe is an oxygen-sensitive, near-infrared emitting macromolecule. It consists of two key functional components. So the first is the large conjugated ligand that extends phosphorescence emission spectra to the near region. This is going to allow light penetration for deep tissue imaging. And then the phosphorescence emission is actually quenched by oxygen in normal tissues, but it turns on at the reduced oxygen level in tumor tissues. And so this provides hypoxia specificity of imaging with minimal background interference. And so here they're able to show this in two different cell lines by changing the oxygen levels. They can turn this imaging probe on. They also show this really nicely in vivo studies. So they inoculate the tumor cells under the armpit area, and then they administer this probe. They track over time. They find the optical imaging time. The signal peaks around 24 hours with very little background. And they validate this with ex vivo imaging. So you can see it's very specific signal only in the tumor, very little to no signal in the other healthy tissues. Excitingly, they're also able to detect metastases with this probe. And so in this model, they're using the same probe. They injected cells into the foot pad and they inject their probe and they find quite a small micromet in the lymph node, actually. They're able to remove the node and they see it ex vivo imaging. And then they did some staining to confirm that both HIF1 is expressed in both the primary tumor as well as the metastasis. There are, like the other modalities, there are, of course, limitations to be considered. The most important one I would say is that hypoxia typically occurs in areas with diminished perfusion. So this is something that really, I think is important, especially for bioluminescence imaging, is this relies on substrate delivery. So you want to make sure that your signal is representative of true hypoxia and not just a limit of perfusion. Some reporters do require cofactors to produce light, such as oxygen and ATP. So again, something to consider if you need oxygen. This may limit your ability to accurately image, I guess, what you're trying to. And then, of course, as with all optical imaging, you need to remember there's going to be a limitation on the depth of the tissue that or the cells that you're trying to image. The more surface level, the better. You're going to have more tissue attenuation and more light scattering at deeper tissues. More recently, groups have started to use live cell imaging. So why? Because really, I think people have realized it can kind of harm or impact your research findings when you're constantly removing your cells from their various environmental conditions. And so this allows you to actually image your cells while maintaining the relevant physiological conditions within either a hypoxia workstation or some sort of chamber. You can visualize hypoxia happening in real time with something like switches or biosensors, or you could confirm hypoxia prior to animal injections. So this can be a really inexpensive and rapid throughput way of validating your in vitro work prior to moving on to in vivo studies. And this can be done both with bright field and fluorescent imaging. So there's lots of options out there for these systems. In terms of bright fields, you could imagine you could do something like just exposing the cells to different levels of oxygen or a certain time frame, and basically assessing things like proliferation or growth rates. Whereas with fluorescent imaging, you have a little bit more flexibility in terms of how you exploit the hypoxia. So similar to what I just showed with the optical imaging, you could do something like they did in this paper where you have imaging genes that are responsive to hypoxia. So this group shows not only different levels of oxygen, but they monitor it over time. And they have two different imaging genes that turn on under different conditions. So there's lots of options here. And this is going to be more at the cellular level. Okay, as we start to near the end of today's webinar, I wanted to highlight this study from Zaver Bourgeois's group at Hopkins. This employs a number of the technologies we discussed today, and I feel it's important to remember that sometimes multiple strategies are needed to answer the question of interest. And so in this study, they exploited the HRE to study the relationship between hypoxia and vascularization using MRI and optical imaging of it was a human tumor xenograft model derived from tumor cells that stably express an inducible GFP. So again, under the regulation of the HRE promoter. They validated the relationship between fluorescence and hypoxia, both in culture and also using the oxylite fiber optic oxygen probe. So they inserted this into both fluorescing and non-fluorescing regions of the tumor under image guidance. So you can see in 2 of the hypoxic regions, they actually get a reading of 0 compared to the peripheral oxygenated regions or the muscle control tissue. They're getting a reading between 42 and 47. They then show distinct vascular and fluorescence distributions detected within the tumors using MR. And so they show low vascular volume regions measured by MRI were associated with high fluorescence or high areas of hypoxia studied with GFP, as well as high permeability. Okay, so how do you choose just one modality with all of these options? Well, I'm hopeful this diagram will help, or at least be a good starting point for those who are interested in choosing one. On the left, you have different degrees of hypoxia in terms of spatial resolution. So from visualizing individual vessels or cells at the top, all the way down to more of a regional hypoxia, where you could consider this either regions of hypoxia throughout a tumor or throughout different parts of the mouse body. Now at the top, we have microscopy or live cell imaging, for example. This is more of a probe-based approach. You could also use reporter gene, but essentially here you're looking at, the resolution you're looking at is single blood vessels or the cellular level using either dyes or optical probes. And this is considered more of a direct way of measuring hypoxia. Now, photoacoustic and ultrasound, we didn't talk about today, but you can image the vasculature using Doppler and or microbubbles with ultrasound, and vasculature and oxygen saturation for photoacoustics. And additionally, both modalities have reporter genes. For MRI methods, these are considered more indirect as you're looking at measures of perfusion, as we talked about earlier, and not hypoxia directly. However, again, there are MRI reporter genes that could provide a different level of sensitivity. Bioluminescence, again, is going to be more indirect because it relies on either gene expression or transcription, essentially, to happen to be able to get that measure of hypoxia. And PET, so while PET has the lowest spatial resolution, it is considered more of a direct method as the probes target markers of hypoxia. So this is kind of a good starting point. I think you really need to think of what you're trying to get out of the study. So what questions you're trying to answer, and the degree of hypoxia that you're looking to kind of visualize or characterize in your model. Okay, we covered a lot of information today in a short period of time. In summary, non-invasive imaging can be extremely valuable for assessing the degree and the distribution of tumor hypoxia over time. This goes for both clinical and preclinical studies. While hypoxia imaging is, a rapidly growing field, its relevant applications continue to evolve. But the three most important areas of focus right now in regards to cancer research are, the ability to identify tumors that are likely to respond to treatment, mapping the extent or degree of hypoxia for treatment planning, and then monitoring treatment strategies designed to modify or alter tumor hypoxia. And to finish, I just wanted to mention again that the focus of today's webinar was on cancer, but we do know that hypoxia plays a role in a wide range of other diseases. And so identifying hypoxic tissue could really have therapeutic implications for multiple disease states. This could include stroke, metabolic diseases, respiratory diseases, and many more. And so for any of you out there that may be studying something other than cancer, I encourage you to reach out, and I'd be happy to talk further about how some of the techniques that we talked about today could be easily modified for your specific research application. And with that, I will pass it back to Gabe for a quick poll question, and then we will hopefully have an opportunity for some questions. Thank you.
Thank you, Katie, for the great presentation. Before we move on to the Q&A section, we would like to ask another set of poll questions, and the questions are the following. Oops, my apologies. I'll just relaunch that. Okay, the questions are the following. Would you like someone to reach out following the webinar to provide further information? And would you like to see other webinars like this one? We'll just give everyone a few moments and we will move on to the Q&A section. We'll just wait a few seconds more. Okay, thank you everyone for answering the poll questions. We will now move on to the Q&A section. And Katie, you mentioned that for optical imaging to work, you need oxygen. So how come in the theranostic study showed they were able to get signal under hypoxic conditions?
Yeah, so that's a good question. It really depends on which luciferase you choose. So as I mentioned, certain luciferases require different cofactors to produce signal. Some of those do require oxygen. So the most common one, firefly luciferase, requires both oxygen and ATP to produce signal. So that one you may want to consider using an alternative, I would say. Other types of luciferases, for example, renylluciferase, has different cofactors. So that's something to consider when you're planning your study. That being said, there are many groups that do still use firefly luciferase, and they found that because the reaction requires very little oxygen, there is still enough there to produce the imaging signal. But I think definitely something to consider if you are really focused on a signal that's truly representing hypoxia.
Thank you very much, Katie. The second question is, how do most people validate or characterize their hypoxia work in vitro prior to moving on to animal imaging studies that can be expensive and time consuming?
Yeah, so that's something I didn't have a lot of time to touch on today. But another good question, of course, you want to characterize either your cell line or your drug prior to moving to animal studies. There's a couple different ways you can do it. A lot of people use the hypoxia workstations or chambers that I mentioned just to try and, I guess, induce hypoxia in more of a in vitro or ex vivo way prior to getting to imaging and finding out that maybe your reporter gene isn't turning on or something like that. You can, if you don't have access to one of these chambers, there are also hypoxia mimetics, such as cobalt chloride that you can use. And it's just like a powder, or you can buy it in a liquid form that you can add to your media. And over a certain amount of time, it can mimic the effects of hypoxia. So that's one way to do it. And then if you don't have access to imaging to be able to, or maybe your study doesn't involve an imaging reporter gene, for example, you can also use like gel imaging to check the expression levels of, like, for example, HIF1 or another hypoxia marker to see if they are responding to hypoxia.
Thank you, Katie, for the great answer. There is another question that we have. Is there a certain technique that is better depending on the type of tumor?
Yeah, that's a tough one. I think the short answer is probably yes. But I mean, that's really up to the investigator to try and kind of optimize their model. I think as some of the studies I showed, the data can really differ from model to model. And that's even if you're, say looking at two models of breast cancer or two models of pancreatic cancer. The levels of hypoxia and the characteristics of the tumor can change quite vastly. And this can really change the data in terms of whether you're looking at maybe more of a perfusion-based method or whether you're using something like optical, where you do need oxygen, for example, to turn on your sensor. And so this is something that I think you really need to consider from each model that you're working on. And as I showed in the last study, I don't think a multimodal approach is a bad idea, at least in the beginning, or maybe for a pilot study, just to get a really good handle on the tumor microenvironment of your specific model and what the data you can kind of expect out of that, depending on the vasculature.
Okay, thank you very much, Katie. There's another question that we have is, do you think these hypoxia imaging can be used for kidney cancer, especially using PET for orthotopic implantation?
Yeah, I would say absolutely. I think you would have to carefully think out which tracer makes the most sense, again, depending on sort of the biological question that's of interest. But yeah, I do. You would want to really carefully think out the clearance of the tracer because you're focused on the kidneys. The kidneys I know can be a little tricky because of the background and how long it can take to clear out. But yeah, I think that's totally feasible.
Okay, thank you very much, Katie. I want to let everyone know that um, there are a couple of questions that we're receiving that unfortunately we cannot answer for the moment. We will answer them offline. You will be receiving a transcript and we'll get in touch with you directly. So I want to let everyone know that we have reached the end of our session for today. And to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of our webinar, we will be sure to answer any questions in the written transcripts, and we will work to get this out to you over the next few days. I'd like to thank Dr. Parkins for the great presentation today, and I trust that we have been able to provide you with a better understanding of the different modalities that can be used to image hypoxia. If in the days and weeks to come you have further questions about the modalities discussed today, I encourage you to reach out to us here at Syntheca, and we will be happy to discuss further. Thanks again to all of you for taking time out of your day to attend our session, and we look forward to seeing you at the future Syntheca event. Have a great day, everyone.